Today, I would like to talk about effects of locally administered human Muse cells on bladder inflammation, overactivity, and nociception in a chemically induced interstitial cystitis-like rat model. This is my COI. This slide shows the characteristics of Muse cells. Muse cells can be collected as cells positive for CR3 in mesenchymal stem cells. Telomerase activity is very low in Muse cells compared with ES and IPS. Muse cells also have the ability to home into damaged tissues through the S1P-S1PR2 axis and to spontaneously differentiate into tissue constituent cells, representative of all three germ layers, which is not seen in ES and IPS. Muse cells have no immunogenicity due to the expression of HLA-G, allowing them to be directly administered without HLA matching or immunosuppressant treatments. 7 clinical trials were currently conducted by the Life Science Institute, Inc. Thus, we investigated the effects of human Muse cells on bladder tissues, function, and nociceptive behavior in a chemically induced HANA-type interstitial cystitis-like rat model. This slide shows the protocol of this study. Human bone marrow-derived mesenchymal stem cells were purchased from Lonza. CR3-positive Muse cells and CR3-negative non-Muse cells were collected by fluorescent-activated cell sorting. We used the 4 groups of animals, including control, vehicle, Muse, and non-Muse cell. In separate groups of animals, the bladder was harvested at 1 to 4 weeks after the implantation of Muse or non-Muse cells to detect the survival. There were significant increases in urinary frequency in the vehicle and non-Muse groups, but not in the Muse group compared with controls in metabolic cage studies. In cystometrograms, bladder capacity was significantly decreased in the vehicle and non-Muse groups, but not in the Muse group compared with controls. Significant increases in nociceptive behavior and expression of C-force were also observed in the vehicle and non-Muse groups compared with controls, whereas these significant changes were not seen in the Muse group. The HE staining showed that severe edema in the bladder epithelium lamina propria with obvious inflammatory cell infiltration was seen in the vehicle and non-Muse groups. Significant increases in expression of TNF-alpha were also observed in the vehicle and non-Muse groups compared with controls. In addition, a larger number of Muse cells survived in the urothelial basal layer and lamina propria compared with non-Muse cells until 4 weeks. The limitation of this study is that the effects of intravenously administered Muse cells on bladder inflammation and function, and the differentiation of implanted Muse cells were not examined. In addition to cell differentiation, paracrine factors with pro-angiogenic and anti-inflammatory effects should also be considered for the repair of damaged tissues and functional recovery. So further studies are needed to clarify these points. In conclusion, the Muse cell therapy could be a promising modality to treat HANA-type interstitial cystitis. Thank you for your attention.